Hi everyone, welcome back to the channel The Science of Time. This is Pratikshya Paul, a third year MBBS student, and today we are going to discuss about the histology of respiratory system and hepatobiliary system. So without any further ado, let's get into the world of blues and pinks. So let's start with the respiratory system first. In respiratory system, I am discussing three slides, that is of trachea, lungs and epiglottis. Out of the three, trachea is the most important slide. The other two are not asked that often in exam. So let's start with the respiratory system. The respiratory system consists of two parts, that is the conducting part and the respiratory part. The conducting part includes the nasal cavities, pharynx, trachea, bronchi and the intrapulmonary continuations. The respiratory part includes the lungs and alveoli. So what is the function of the conducting part? It is the passage of air and the conditioning of air. Conditioning of air means to maintain the humidity and the temperature of the inspired air. And what is the function of the respiratory part? It is to exchange of oxygen and carbon dioxide between the blood and the inspired air. Okay, now let's come to the slides. First is the trachea. So what is the lining epithelium of the trachea? It is pseudostratified ciliated columnar epithelium and along with the presence of goblet cells. And the lamina propria is seen underneath the epithelium. The submucosa contains the serous and mucous glands that produce the mucus and presence of hyaline cartilage. This is the most important characteristic feature of trachea, that is the presence of hyaline cartilage. Trachealis muscle can also be found in the substance of trachea, but this is not an important feature because you cannot identify it always. So what are the most important characteristic features of trachea? That is the presence of pseudostratified ciliated columnar epithelium and the presence of hyaline cartilage. Also, the pseudostratified ciliated columnar epithelium is also known as respiratory epithelium because it is mostly found in the respiratory system. Okay, now let's come to the slides. Can you see these, the pseudostratified ciliated columnar cells? This is where the general histology comes into play because अगर तुमने general histology अच्छे से पढ़ी होगी तभी तुम यहाँ पे आके pseudostratified ciliated columnar epithelium identify कर पाओगे अच्छे से. Because the nuclei are present at the different level, it gives rise to a stratified appearance, but it is not a stratified epithelium. That's why it is called pseudostratified epithelium. Okay, so beneath the pseudostratified epithelium, the lamina propria is present. This is the lamina propria, and these are the serous and mucous glands present. These are the serous and mucous gland present in the lamina propria. This you can identify very well. This is the hyaline cartilage, and how can you identify hyaline cartilage? By the presence of this basophilic matrix. That is the most easy way to identify hyaline cartilage, presence of basophilic matrix. Okay, so this is the original slide picture. This is the original slide picture. You can also identify here the basophilic matrix. This is the pseudostratified ciliated columnar epithelium, and these are the glands present in the lamina propria. So you can now easily identify the slide of trachea, and this is the picture from the diffuse atlas which you have to draw in your practical manual. So the most important characteristic feature that you have to remember is pseudostratified ciliated columnar epithelium and the presence of hyaline cartilage. So this is everything we have to discuss about the slide of trachea. Now you can easily identify the slide of trachea. Okay, so now let's go into the next slide, that is the slide of lungs. So what are the characteristic feature of lungs? Alveolar sac is lined by simple squamous epithelium, presence of intrapulmonary bronchus with islands of cartilage and smooth muscle, and the presence of alveolar ducts. So simple squamous epithelium तुम आइडेंटिफाई कर पाओ या ना कर पाओ इतना कुछ मायने नहीं रखता, but the presence of intrapulmonary bronchus and islands of cartilage and alveolar ducts you have to identify because these are the main characteristic features of the slide of lungs. So can you appreciate here? This is the island of cartilage that is present, and these are the smooth muscles present in the slide, and these are the intrapulmonary bronchus that are present, and the open spaces that you are seeing, the open spaces, these are the alveolar ducts. Okay, now you can easily identify the slide of lungs from these three things: the island of cartilage, the intrapulmonary bronchus, and the alveolar ducts. Okay, and this is the image from the diffuse atlas that you can draw in your practical manuals. सो वन थिंग आई वांट टू टेल हियर, ऐसा हुआ भी है कि बच्चे गलत करते हैं कि इसमें उनको एडिपोज टिश्यू दिखता है क्योंकि बहुत सारा खाली स्पेस है, तो एडिपोज टिश्यू भी ऐसा ही दिखता है। अगर तुम शुरू में आइलैंड ऑफ कार्टिलेज और इंट्रापुल्मोनरी ब्रोंकस आइडेंटिफाई नहीं कर पाओगे तो तुम्हें एल्वियोलर एडिपोज टिश्यू जैसा ही दिखेगा, क्योंकि वो भी ऐसे खाली स्पेस जैसा होता है, एक पिंक कलर का बॉर्डर होता है और एक एसेंट्रिक न्यूक्लियस होता है। तो इसीलिए बोल रही हूँ, आइलैंड ऑफ कार्टिलेज एंड इंट्रापुल्मोनरी ब्रोंकस अगर तुम आइडेंटिफाई कर लोगे तो तुम एडिपोज टिश्यू के साथ ऐसे कंफ्यूज नहीं करोगे। ओके, सो दिस इज एवरीथिंग यू हैव टू नो अबाउट द स्लाइड ऑफ लंग्स, नथिंग मच टू डिस्कस हियर। नाउ लेट्स कम इनटू द स्लाइड ऑफ एपिग्लोटिस। So what is the lining epithelium of epiglottis? Epiglottis has two kinds of epithelium, one towards the lingual mucosa and other towards the laryngeal mucosa. So what is the epithelium towards the lingual mucosa? It is the stratified squamous keratinized epithelium, and towards the laryngeal mucosa it is the pseudostratified ciliated columnar epithelium, because towards the larynx it is the respiratory side, so the respiratory epithelium is present towards the laryngeal mucosa. And the lamina propria contains the serous and mucous glands, same as trachea, and the major difference from trachea is the presence of elastic cartilage. In trachea there was hyaline cartilage, but in epiglottis it is elastic cartilage. You can also remember that E for E, because epiglottis start from E and elastic cartilage start from E, तो तुम्हें याद रखने में आसानी होगी। Okay, so दिस इज द कैरेक्टरिस्टिक फीचर ऑफ एपिग्लोटिस। तुम्हें दो एपिथेलियम आइडेंटिफाई करना है एंड इलास्टिक कार्टिलेज आइडेंटिफाई करना है। ओके, नाउ लेट्स कम टू द स्लाइड्स। सो यू कैन इजीली आइडेंटिफाई द epithelium here. This is the pseudostratified this is the stratified non-keratinized epithelium, and this is the pseudostratified ciliated columnar epithelium. Again here you can see the same thing. This is the stratified squamous non-keratinized epithelium, and this is the pseudostratified epithelium, and you can identify this is the elastic cartilage that is present. And towards the lingual mucosa you can also find the papillas that are present because it is towards the tongue, and the serous and mucous glands are present in the lamina propria. Okay, this is the original slide picture. This is the stratified squamous non-keratinized epithelium, and these are the pseudostratified ciliated columnar epithelium, and this is the elastic cartilage that you can identify. Okay, and this is the picture from the diffuse atlas that you have to draw in your practical manuals. So this much about epiglottis. The main feature is to identify two types of epithelium and the presence of elastic cartilage. These are the differentiating features from the trachea, because these two slides look somewhat similar under the microscope. तो तुम पहली नजर में देखोगे तो तुम्हें नहीं पता चलेगा कि ये ट्रैकिया है या एपिग्लोटिस है। ध्यान से देखने से ही पता चलता है कि एक एपिथेलियम है या दो एपिथेलियम है। बाकी तुम कार्टिलेज तो इजीली आइडेंटिफाई कर पाओगे, बिकॉज देयर वाज हाइलिन कार्टिलेज इन ट्रैकिया व्हिच इज बेसोफिलिक इन नेचर, एंड देयर इज elastic cartilage in epiglottis. Okay, so this is the 3 slides of respiratory epithelium that I am discussing here. Other things are not important in respiratory system. These are the most important slides and most often asked in the exam. Now let's start with the hepatobiliary system. In hepatobiliary system I am discussing 2 slides. One is liver and another is gallbladder. Liver is an important long question in anatomy, and along with the long question, as a part of the long question, the microanatomy of liver or the histology of liver can be asked. There you have to draw only the diagram. You don't need to write the characteristic feature until it is asked. But if as a histology slide exam, you have to draw the diagram and you have to write the characteristic features also. Okay, so let's start with the slide of liver. What are the characteristic features of liver? So it shows many hexagonal areas called as hepatic lobules. The lobules are separated by connective tissues. The lobule has a central vein at the center and a portal triad at the corner. And the portal triad contains a portal vein, a hepatic artery, and a bile duct. The cords of hepatocyte pass from the central vein to periphery of the lobule, known as the hepatic lamellae. Okay, so these are the important features of liver. You have to remember the hexagonal shape of the hepatic lobule, the presence of central vein at the center and the portal triad at the corners. And you have to remember the constituent of the portal triad that is often asked in the viva, that is the portal vein, the hepatic artery and the bile duct. Okay, so you can easily identify the slide of liver from the hexagonal shape of the hepatic lobule. So it is often asked as a spotter also, because बहुत जल्दी identify कर पाओगे तुम कम time में. Okay, so you can see here, this is the hexagonal lobule, that is the hepatic lobule present. Okay, and this, the space between the two lobules, these are the connective tissue present between the two lobules, and this is the central vein present at the center, and these are the portal triads present at the corners. Okay, and the portal vein, the hepatic artery and the bile duct can also be appreciated under the microscope. Okay, and these are the hepatocytes that are radiating from the central vein towards the periphery. The whole thing is known as the hepatic lamellae. Okay, you can see here also, this is a more magnified image. Okay, so this is the central vein and these are the hepatocytes. These are the hepatocytes present, and these are the cords of hepatocyte. The core of hepatocyte is known as the hepatic lamellae. These are the hepatic lamellae. And the open spaces that you are seeing, these open spaces are known as sinusoids. These are the hepatic sinusoids. Okay, and this is the image from the Dufour's atlas. So this much about the slide of liver. You can easily identify the slide of liver. It is the easiest slide, because एक बार तुमने हेक्सागोनल स्ट्रक्चर देख लिया तो कंफर्म है कि ये लिवर है, बिकॉज ऐसा और कोई स्लाइड नहीं होगा. ओके, गलती नहीं करना है इसमें. Now there are some questions associated with the viva of slide of liver. They are asked in the viva often, like the space of mole, what is space of Disse, what is a portal lobule, what is the difference between portal lobule and portal acinus. So I am discussing it briefly here. So what is a space of mole? It is a potential space between the Gleason's capsule and the portal triad. It is a potential space between the Gleason's capsule of portal triad and the hepatic plate. You know the capsule of liver is known as Gleason's capsule. So जो portal triad का capsule होता है और जो hepatic plate होता है, दोनों के अंदर जो gap होता है उसको बोलते हैं space of mole. Understood? Now let's come to space of Disse. What is a space of Disse? It is the space between the endothelial lining of sinusoid and the surface of hepatocyte. Suppose this is a hepatocyte and this is an endothelial cell. So the space between the hepatocyte and the endothelial cell, this is known as the space of Disse. This is the space of Disse. Okay, it is an important question. It is often asked in viva. What is space of Disse? Now let's come to the portal lobule and portal acinus. So what is a portal lobule? It is a triangular area of liver tissue which with portal triad at center and the central vein at each corner. You can understand this by seeing into this diagram. These are three hepatocytes that I have drawn, and this is the central vein that is present at the center of each hepatocyte, and this is a portal triad present at the corner of the three hepatocyte. Okay, now if you join the three central vein via imaginary line, then there will be a triangular area. This triangular area is known as portal lobule. This is known as portal lobule. Okay. Now what is portal acinus? Same. This is hepatocyte. This is hepatocyte, and in the center the central vein is present, and at the two corners there are two portal triads. Now connect the four structure by an imaginary line, and there will be a diamond shaped structure. This diamond shaped structure is known as the portal acinus. Okay. So now you know the difference between portal lobule and portal acinus. Another important question that is asked is the what is the phagocytic cell of liver, and it is known as the Kupffer cell. Okay, so these are the important questions that are asked in the histology viva of liver. Now let's come to the slide of gallbladder. This is everything you need to know about liver. So what are the characteristic feature of gallbladder? It has an inner mucosa, a middle fibromuscular and an outer serosal layer. The lining epithelium is tall columnar cells and the mucosa is highly folded. So this is where the confusion arises. Because the mucosa is highly folded, it gives rise to appearance of villi. But it is not a true villi, it is the mucosa that is highly folded. So you have to differentiate it from the small intestine, because in small intestine there is presence of true villi. Okay, so how do you differentiate the slide of gallbladder from the small intestine? It is differentiated from small intestine by the absence of villi, the goblet cells, submucosa, and the proper muscularis externa. So the villi is absent in gallbladder, goblet cell are also absent in gallbladder, and the submucosal layer is absent also. This is an important part. You have to remember the submucosal layer is absent in gallbladder, and in other parts of GIT it is present. It is often asked by the examiner. Also, the proper muscularis externa layer is not present. The muscularis layer is not well developed in gallbladder. So, in place of muscularis layer, there is a fibromuscular coat present in the gallbladder. Okay, now you can easily differentiate the slide of gallbladder from the slide of small intestine. Now let's come to the slides. You can easily appreciate the tall columnar cells here. Gallbladder, in gallbladder the tall columnar cells are easily appreciated. जितना अच्छे से columnar cell गॉल ब्लैडर में दिखता है ना, उतना अच्छे से कहीं किसी और स्लाइड में नहीं दिखता. So you can easily appreciate the tall columnar cell, and you can see the mucosal folds. These are highly folded mucosa. So this may give rise to the appearance of villi. Okay, and this is the original slide picture. You can appreciate the presence of tall columnar cells, and these are the highly folded mucosa that is present, and this is the lamina propria, and this is the fibromuscular coat present in the gallbladder. Okay, and this is the diagram from the Diffier's atlas. So this is pretty much about the slide of gallbladder that you have to know. You have to differentiate it from the slide of small intestine, and what are the differentiating feature in gallbladder and small intestine. This is also asked in exam. So this is pretty much about the hepatobiliary system. I have discussed the slide of liver and the slide of gallbladder also. So I have discussed the important slides of respiratory system and hepatobiliary system in this session. That's it from my side for this video. In the next session I will again discuss the remaining systems of histology. Till then, keep studying, keep watching. Thank you so much for watching.